Hello students, welcome to EPG Pathshala. I am Dr. J. Arunachalam from Sastra University. Today I am going to talk on the module named Introduction to Systems Biology from the paper Bioinformatics. The specific objectives of this module is to introduce the fundamental concepts behind systems biology, to explain the various properties of biological process at a system level, explain the concept of a systeome, and to relate the systeome to genomes and proteomes. The final objective is to provide an overview of some important applications by systems modeling. The progress in molecular biology have led us to have a very detailed understanding of biological process today. Today, we have a molecular level understanding of almost of many important biological process. Like say for example, we have the cell division process. We can say at a molecular level what happens, what are all the proteins that are involved in cell division, number one, and number two, what are all the expression pattern of the genes that are involved in cell division. And then we can also say at a molecular level how these proteins interact in order to bring cell division. The same could be said for any biological process. Let us say DNA replication even. We can say at a molecular level we can give an understanding like this is how the DNA unwinds. These are the proteins that come and bind to the DNA while it is unwinding. This is how the polymerase comes and attaches itself to replication. Or these are the different subunits of the polymerase. This is how the actual replication process is going on. This is how the reaction is being catalyzed. So today, with the advances in molecular biology, we are able to give a very detailed molecular level understanding of a biological process. By this, we are able to identify genes, we are able to identify their functions, the mode of regulation of these genes or biological phenomena, the expression pattern of genes in different biological phenomena, the interaction between the components can all be studied by molecular biology experiments. So the advances in molecular biology reveals the details of the components of a biological system. That is very important to understand. So whatever advance that we have in molecular biology only reveals the details of the components of a biological system. Now we have to bring these advances to the next level of understanding. We need to integrate all these components in order to obtain a larger level of understanding, which is the system level of understanding. So systems biology can be defined as a field of biology that aims at system level understanding of biological systems. See, we have to understand that it is not a simple process of adding up all the components that we have already understood. So when we try to add up different components at a molecular based on their molecular level understanding, and when we try to bring them together to give a systems level understanding, a simple addition of the components won't be enough. As we always say, the whole is much bigger than the addition of the parts. So when we try to bring them together, when we try to bring the components together, it brings in its own problems that has to be addressed, and then only by addressing them we will bring the molecular level understanding to the next level, which is the system level understanding in biology. And to achieve this, it involves the application of several methods like experimental methods, theoretical methods, computational methods, and several modeling techniques. An overview of systems biology. Systems biology requires a problem-specific approach. It is a hypothesis-driven research approach, and it eventually aims to model cells, organisms, and perhaps humans in future, the system level in a consistent framework of basic principles. So cells, organisms, and species are considered to be at the system level, and we should be able to model them in a consistent framework of basic principles for which the data is derived from the experimental procedures. So Here you have a set of experimental data based on which you create a hypothesis. You simulate that, see how well they fit into the hypothesis, try to model the system based on components, and then once again see how well it fit into the experiments and the results. So basically it is cyclic, it is hypothesis driven, and eventually it aims to give a system level understanding of the observed phenomenon in biological systems. So as I told you before, it involves generation of a model representing the biological phenomena under study. So what is a model? A model is a representation of a hypothesis which could be tested computationally, and it should also be supported experimentally. So computational simulations are done to check the adequacy of the models based upon their agreements with experimental results. So we create a model, simulate that, see how well it fits with hypothesis as well as the experimental results. The predictions can also be made based on adequate model. So once we have an adequate model, we will be able to make appropriate predictions from the model, which once again these predictions can also be tested experimentally. So the interaction between the various disciplines, genomics, computation, data analytic procedures, and the technologies that we have here are described in this figure. So the entire hypothesis driven research can be explained here. So we have biological knowledge, and we also have contradictory issues. There are computation methods, that computation and data analytic procedures, the data and hypothesis driven modeling. We can do simulations, then system analysis and theory formation. We make predictions, and based on the predictions we once again go back to the experiment and the experimental device development, not only experiment design but also the device development. We do wet lab experiments and then data analysis of the experiments, which once again adds more knowledge to the biological phenomena, and this could go on as a cycle until we are able to explain any given biological phenomena at a systems level. After having an overview, we need to know about the structure of the system and how to identify them. The system structures include the network of gene interaction, signal transduction networks, metabolic pathways, and several other interaction networks. So it also several other underlying mechanisms of interactions comes in as networks. These could be the system structures. Some examples for system structures. So for example, in a developmental process, we require the structural relationship among cells. So these interaction could be at various level. So for example, we take the development of organs in Drosophila. So let us say the development of eye, or development of antenna, or development of the limbs in Drosophila. This process, in order to model this process, we need to know the relationship between cells at various stages of embryogenesis. So we need to know the relationship among different types of cells. Detailed information about cell-cell contacts, etc. are needed to model this process. Whereas if you take a different process like some intracellular process, like a vesicular secretion, or like chromosomal segregation. So these information, to model these, we need information about intracellular structures, gene regulatory networks, etc. that lead to this intracellular process. So the structure of systems which we described before, it could be anything, it could be an extracellular process or intracellular process, but we need to have a complete understanding of the networks, that the components that are involved in the network in our system of interest. After having an understanding of that, we need to identify the structure of the systems. So how do we identify the structure of the systems? That is the next important question. A gene regulatory network, as an example, we can take that gene regulatory network as an example, and here we need experimental data on all possible components of the network, and then we need the function of each component, their interactions, and all the associated parameters for the network is also important. The inference of the existing experimental results should also allow us to predict hitherto unknown genes and their interactions, which could later on be verified by experimental procedures. The entire process of this identification can be the entire process of this structure analysis can be divided into two steps, which is the network structure identification and then the parameter identification for the identified network. Network structure identification. There are two generally used approaches for network structure identification. The bottom-up approach, it brings together several independent experimental data. For example, the early embryogenesis data of Drosophila, organ formations such as eye, wing, etc. in Drosophila, and lytic lysogenic cycle in lambda phage, etc. So it brings together all independent experimental data and tries to give us a whole system-based picture. The second approach is the top-down approach, where high throughput data such as transcriptomics data, microarray data could be used to identify the network structure. These methods use computational procedures such as clustering, heuristics, genetic programming, etc. to identify the structure. For example, modeling of yeast cell cycle based on microarray data could be an excellent example for the top-down approach of network structure identification. After identifying the network structure, it is important to identify the parameters that are associated with the network structure. So, the parameter identification is to match the computational results against the experimental results. The identified network along with its parameters will be used for simulating a quantitative analysis of the system's response. The parameter identification can be considered as an optimization problem, since what we have is a network and then some experimental data which relates to the network. So now we have to identify a set of parameters for this network that relates, that agrees with the observed experimental result. So it is an optimization procedure. So several optimization procedures and algorithms such as simulated annealing, genetic algorithm, etc. can be used for parameter identification. But we should remember that there could be multiple parameter sets which can simulate same experimental results. So it is like identifying several multiple minima in our multiple minima or multiple maxima in our optimization procedure. So there could be multiple parameter sets for a network whose response upon simulation will match with the experimental results. The system behavior analysis. Analysis of the dynamics of identified network structure is the next step. After identifying the network structure, we need to analyze the dynamics of the identified network structure. Why should we do this analysis? It will help us to answer some important questions like how will the system respond to changes in environment? For example, you have modeled cellular system, and if you want to see what happens to the system or upon introduction of a new nutrient to the cell or any other stimuli, it could be any other stimuli. Once we have modeled an appropriate biological phenomena, it will be useful for us to study what will happen to the model if something external is introduced or if something is changed in the model. So that is a primary question that can be answered here. So how does a system respond to changes in the environment such as nutrition or other stimuli? The second important question is how does it respond to damage such as a DNA mutation? So how does the system respond to any type of damages? So extensive computer simulations and corresponding theoretical analysis are required to analyze the system behavior. The simulation software should be efficient enough to handle differential equations in the case of high concentration of proteins and stochastic process as in the case of very low concentration of proteins. So whatever system analysis we do or whatever simulation that we do should be efficient enough to handle deterministic procedure like solving ODEs, the differential equation, or it should also be efficient enough to handle stochastic processes. So moreover, it should also be able to handle higher orders such as chromosomes, heterochromatin, etc. if the need arises. So these are all the requirement for whatever procedure that we use to perform a system behavior analysis. A set of software systems for systems modeling need to integrate the following based on the requirement that was explained in the previous slide. So it needs to have efficient database for handling experimental data, an efficient cell, tissue or any system simulator, parameter optimization procedures, bifurcation and system analysis, hypothesis generation and experimental planning. It should also have efficient data visualization procedures. So all these requirements should be integrated together to perform an excellent system behavior analysis procedure. Relationship among software tools. So this flowchart gives us the relationship between different software tools and how they could be integrated together to perform system analysis, which we have explained earlier. So for example, you have system measurements. To get the experimental data, we need an appropriate interface for that, and then experimental data should be present in appropriate database so that the simulator can seamlessly interact with the database and obtain the data for simulation as and when necessary. Similarly, we can have a genome proteome database for which we have to associate the experimental values that interacts with system software database, parameter optimization module. Everything has to interact with the visualization module. So and there should also be efficient hypothesis generation, experimental planning module. Finally, everything is related to a system analysis module which does a bifurcation analysis, flux balance for example. So several analyses should also be done. So this flowchart is an example of how well system level integration for performing a system analysis should be done. Workflow on software tools. This flowchart explains the different software tools and their interactions in systems modeling. For example, you have expression profile data, two hybrid data, RT-PCR data, etc. Describing the expression. Similarly, we have gene regulation network, metabolic cascade network, signal transduction networks. They all can be given as input for software components like parameter optimizer, a simulator, hypothesis generator. And once again, this should aid in creating appropriate experimental design and assistance systems, which in turn leads to several other experimental plans, and then biological experiments are done. This data once again goes back into the original starting point where we can do it as a cycle. So here, this, as I explained earlier, this flowchart explains the interaction between various software tools that has to be integrated to perform an appropriate systems analysis. The control methods. These control methods are mechanisms that control the state of the system. To obtain a higher level of robustness and stability, the following aspects of engineering systems are extensively applied: the system control, redundancy, modular design, and structural stability. The next important concept that we have to understand is how the system control is achieved. Normally, there are two important methods of system control, which is the feed-forward control and the feedback control. A feed-forward control is an open loop control where a set of predefined reactions are triggered upon a certain stimuli. Like for example, signal transduction mechanism. So you have a stimulus which triggers a set of reactions which could be considered as a feed-forward control. Similarly, there is a feedback control which closes the loop of a circuit. So feedback control closes the loop of a circuit. It modulates the input of a circuit by sensing the ratio between the desired output and the actual output. So there might be a desired output and an actual output, and the ratio is always crucial to decide on whether the reaction should proceed or not. So if the ratio tilts to the other side, the same output can go back and stop the execution of the loop. So feedback control closes the loop of a circuit. It modulates the input of a circuit by sensing the ratio between the desired output and the actual output of the process. Feed forward and feedback controls. Feed forward and feedback controls are explained here with an example of heat shock response in E. coli. So, E. coli has a heat shock response circuit. Any heat shock in E. coli cells leads to a production of sigma 32 and correspondingly sigma 32 RNA-P haloenzyme, which activates appropriate HSPs, the heat shock proteins. This process is called as the feed forward control and it is explained in the flowchart. Similarly, it also has DNA K and DNA J proteins that detect the misfolded proteins and release the sigma 32 that is already bound to them. So this free sigma 32 activates the transcription of sigma 32 and in turn the heat shock proteins. So this is a classic example for negative feedback control. So E. coli heat response has both feed forward control and feedback control. Redundancy. Another example for redundancy can be given by MAP kinase signal transduction cascade, which is shown in the figure. So here the redundancy is achieved by using multiple pathways towards the same objective. So there are several examples like this where there are 3 or 4 pathways with a single objective, all of them performing the same function. So by this system achieves a redundancy. Here these type of redundancies are normally achieved by duplicated genes or genes with similar function, and it could be built at a circuit level and also such as different signal transduction pathways. So that is an example for redundancy at circuit level. We have different signal transduction pathways which for a single objective. One such signal transduction pathway is given here as an example, which is a MAP kinase signal transduction pathway. These cell cycle genes CLN and CLB have 3 and 6 copies. That is an example of duplicated genes or genes with similar functions. There could be several examples that could be given for this. For example, the SOD gene of several eukaryotic organisms. So there are several types of superoxide dismutases which perform the same function. Copper zinc SOD or the MN SOD. So the function of them are the same. They are proteins with similar functions. Modular design is another organizational setup in any systems. So modularity, it can be viewed as a hierarchical organizational setup for any system. So let us see them according to their hierarchy. The first hierarchy is the component. For the first level among the hierarchy is the components. So they are the genes, the proteins, the metabolites, etc. They can be considered to be similar to the transistors, capacitors and resistors in an electronic circuit. The second level of modularity is called as the device. So in order to give an example for them, let us say the transcription complexes, the ribosomes, signal transduction circuits, etc. These can be considered as devices. So they come as the next level. They could be compared to the gates, flip-flops, etc. in an electronic circuit. And the next level of hierarchy is the modules. These are the large cluster of devices. So earlier we saw what were devices. Now when the devices forms large clusters, so the large cluster of devices from the modules, form the modules. So these, in order to give some examples for these, let us say cell cycle networks, cellular organelles, etc. They could be called as modules. In order to give a relationship with electronics, we can say that CPU, amplifiers, etc. in electronics can be considered similar to that of the modules in systems biology. The final level of modularity comes from the system in itself. It is the assembly of the modules which make up the cell or even an organism in itself, depending on the objective of our it depends on our interest, at what level we want to explain a biological phenomenon. So it is like an entire computer in an electronics system. Structural stability. One example for structural stability, the use of feedback loops, lambda phase circuits, lytic and lysogenic cycle of lambda phase. That example is shown in the figure here. It is the structural stability can be defined as the ability of circuits as a response to certain stimuli or disturbances to resume as one of the multiple attractors. In the lambda phase lytic lysogenic regulatory circuit, the earlier commitment of the circuit is determined by a stochastic process. The feedback loops involving CRO and CL proteins play an important role in the maintenance of the commitment. So the earlier commitment is stochastic here. Once it is committed, the CRO protein, the CRO protein and the CI protein maintains the commitment. So the maintenance of the feedback is determined by the amount of the activator binding to the OR, and the activator itself cuts off the binding to OR if the amount exceeds a threshold. So it acts as an excellent feedback mechanism by which the commitment could be maintained. The Systeome project. The Systeome project is an ambitious project which aims to have a comprehensive data repository of system features similar to that of a human genome project. So in human genome project we have the entire genome sequence. Similarly, this Systeome project aims to get the entire repository of system features in humans or any other organisms. So a Systeome is an assembly of all systems profiles which includes the different genetic variants, the environmental stimuli and responses. So it could be considered as a set of properties of a system which includes its structures, behavior, analysis of the results and the bifurcation diagrams. A human Systeome, the human Systeome project aims to develop a Systeome for a human being. It's like a genome project that we had earlier. So, however, there are main differences. A Systeome is not a static entity. It is an active and dynamic profiling of different system points. That is something that is very important, which we have to understand. Even a genome is not a static entity, but normally in bioinformatics, when we say genome, what we consider is the sequence of the entire genome, which is static. So a Systeome is not like that. A Systeome is a non-static entity. It is an active and dynamic profiling of different system points. The goal of the human Systeome project is to complete a detailed and comprehensive simulation of an entire human cell with a 20% error margin by 2020, and to identify the system's profile of all genetic variations and drug responses, etc. by 2030. That is the goal of a human Systeome project. The relationship between genome, proteome and the Systeome. This relationship is shown in the figure. So genetic variations in the form of SNPs can be expected to play a crucial role in systems modeling. Similarly, the components that exhibit quantitative sensitivity such as cyclins and their responses in cell cycle should also be integrated in order to give a complete picture. So genome data, from the genome data it's possible to obtain the SNP variations and its corresponding influence on the systems should also be analyzed, identified. Similarly, quantitative information of proteins like cyclins and their effect on the system, that information should also be integrated. The figure explains clearly the relationship between various other parameters like that have been explained in the first 2 points in order to obtain an individual Systeome. For example, we need dynamics information by high resolution imaging, expression profiles, protein interaction profiles, etc. Similarly, genome, proteome data, so they are also component of information. And then we have basic model information, the basic structure, the network model, the parameters associated with that. Similarly, there are dynamics information from, for example, the drug sensitivity analysis. All of them should be integrated together and it all forms an individual Systeome. So this Systeome can also help us to identify evolutionarily conserved circuits. So from the sequence data, we are able to identify evolutionary conserved patterns among genome sequences. We are able to interpret evolution based on gene sequences or protein sequences. Eventually, if we have a Systeome, it is also possible to interpret evolutionary system processes in organisms, which should be more meaningful than interpreting evolution by the sequence data alone. Applications of systems biology. Systems biology has very important applications in healthcare, such as simulations of patient models, disease models. It's possible to create disease models, assess disease risks, etc. Similarly, it also has important applications in drug design. Patient-specific drugs and patient-specific dosages of drugs are all possible if you are able to create an appropriate systems model and appropriate simulations which can measure the responses on the models to the drugs that has been introduced. Similarly, advances in regenerative medicine, it can help to advance in regenerative medicine, organ cloning, etc. As one of the application, flowchart describes the drug discovery process and associated systems biology that could be involved in a drug discovery process. So we can have two interacting phases where drug discovery and treatment optimization and hypothesis testing and knowledge discovery. So these two interacting process can aid in efficient drug discovery, as well as it can also aid in efficient patient specific and dosages drug administration. So students, let us now summarize what we have learned in this module, Introduction to Systems Biology. Systems biology is an emerging field of biology which tries to integrate component level knowledge to provide us a systems level understanding of a biological process. Systems biology is a very broad discipline and involves a highly multidisciplinary approaches. Such a process requires development of new software, mathematical modeling procedures, analytical procedures, hypothesis generation, and experimental testing. It has very important medical applications like drug design and administration, organ cloning, transplantation, etc. Thank you very much.